Welcome to the deep dive. Today we're zooming right into the microscopic world inside you, exploring something really fundamental, how your cells react when things get tough. We're talking stress, injury, and eventually even cell death. It's happened constantly. So our mission, our deep dive today, is all about altered cellular and tissue biology. And we're sticking strictly to one key source, Understanding Pathophysiology, 7th edition. Think of this as your essential guide to the absolute basics of what goes right and wrong at the cellular level. We'll walk you through it.
Exactly. And understanding this stuff, it's like getting a backstage pass. It helps you connect the dots between these tiny cellular events and, well, the bigger picture of health and disease. It really is ground zero for pathophysiology.
Okay, right. Let's unpack this then.
Mm-hmm.
So our cells are always trying to maintain this balance, this steady state, homeostasis, right?
That's the goal, yeah. Homeostasis.
And when things push them off balance, maybe a new stressor or demand, they first try to adapt. So cellular adaptation, these are reversible changes cells make to basically avoid getting injured.
Right. And it's important to remember, an adapted cell isn't exactly normal anymore, but it's not technically injured either. It's sort of hanging on, adapting. This could mean changing its size, the number of cells, maybe even the type of cell. But, there's a limit. Too much stress or stress for too long.
And the adaptation fails.
Exactly. Then you're looking at cell injury or even cell death.
Okay, so let's look at how they adapt. The first way is atrophy. Simple enough. Cells shrink. And if enough cells do it, the whole organ can get smaller.
Yeah, and we see this normally, like the thymus gland shrinks as you grow up, ovaries after menopause. Even some brain cells naturally shrink a bit with age. That's physiologic atrophy.
But it can also be pathologic, right? Like if you have a cast on your arm.
Perfect example. That's disuse atrophy. The muscle cells aren't working, so they shrink down. Mechanistically, they're basically slowing down making proteins and speeding up breaking them down. It's cellular housekeeping, getting rid of stuff it doesn't need right then. You might even see little brown spots, lipofuscin, build up from this cleanup process.
Okay. The flip side of shrinking is hypertrophy. Cells get bigger, so the organ gets bigger. This is usually a response to increased work.
Right. Like think of a runner's heart. Those cardiac muscle cells get bigger and stronger from all the aerobic exercise. That's physiologic. Or if, say, someone loses a kidney, the other one often gets bigger hypertrophies to compensate.
But like atrophy, there's a pathologic side too.
Definitely. A classic example is the heart dealing with chronic high blood pressure. The left ventricle, the main pump, has to work harder. So its muscle cells get bigger, thicker. If you could look at figure 4.3 in the text, you'd see that thickened wall. Initially, it helps, but over time,
it becomes a problem.
Yeah, it can lead to the heart struggling, becoming less efficient, maybe even failing. You often see more scar tissue, fibrosis building up in there too. So a clear difference between healthy adaptation and something going wrong.
Okay. Next up, hyperplasia. This isn't bigger cells, but more cells. An increase in cell number due to more cell division.
Correct. This happens when growth factors signal cells to divide, or when stem cells ramp up their output. And often, you'll see hyperplasia and hypertrophy happening together in the same tissue.
And again, there's a normal physiologic version.
Absolutely. Think about wound healing or how your liver can regenerate if part of it is removed. That's compensatory hyperplasia. Or a callus forming on your skin from friction. Hormones drive it too, like the endometrium thickening each menstrual cycle. That's hormonal hyperplasia.
But the pathologic side
Can cause trouble, like too much estrogen stimulation can lead to endometrial hyperplasia, causing abnormal bleeding. Or in older men, benign prostatal hyperplasia, BPH, the prostate enlarges due to hormonal shifts. Figure 4.4 shows this, an enlarged prostate gland potentially squeezing the urethra.
making it hard to urinate.
Precisely. The good news often is that if you can remove that hormonal trigger, the hyperplasia might regress.
Now, the next one, dysplasia, you mentioned, isn't really a true adaptation.
Right. It's sometimes called atypical hyperplasia. It's characterized by abnormal changes in the size, shape, and organization of mature cells. It's not orderly growth.
Where do we usually see this?
It's common in tissues that turn over quickly, like the epithelium of the cervix or the lining of your respiratory tract. If you looked at figure 4.5, you'd see cells that are supposed to be neat and uniform, looking all jumbled and varied in size and shape.
But it can be reversible.
Yes, if the irritating stimulus is removed. But, and this is critical, if those abnormal cells break through the underlying tissue layer, the basement membrane, then it's considered an invasive cancer. So dysplasia is like a serious warning sign.
Got it. Not cancer yet, but could be heading that way. Okay, last one. Metaplasia.
Metaplasia is when one type of mature, differentiated cell gets replaced by another type, often one that's maybe tougher but less specialized.
Like an adaptation to a harsh environment.
Exactly. But it often comes at a cost. The classic example is in the airways of a chronic smoker. The normal cells lining the trachea and bronchi are delicate ciliated columnar cells. They have little hairs, cilia, to sweep mucus and debris out.
Okay.
In a smoker, these can get replaced by stratified squamous cells. Think tougher, flatter cells, more like your skin. Figure 4.6 illustrates this swap. Now, these squamous cells can handle the smoke irritation better,
What?
but they don't have cilia and they don't make protective mucus. So the smoker loses that crucial cleaning mechanism, making them more prone to infections. It happens because the stem cells in the area get reprogrammed.
So adaptation is the first response, but it's not always enough. What happens when cells can't adapt or the stress is just too overwhelming?
That's when we cross the line into cellular injury. This can still sometimes be reversible. The cell might recover if the stress stops, but if it's too severe or prolonged, the injury becomes irreversible, and that leads to cell death.
What kinds of things go wrong at the biochemical level during injury?
There are several common themes often happening together. Number one is ATP depletion. The cell runs out of energy. Then there's damage from reactive oxygen species, those unstable molecules. Calcium levels inside the cell can go haywire. The mitochondria, the powerhouses, get damaged. The cell's outer membrane integrity is compromised. Proteins might misfold, and DNA can get damaged. Table 4.2 lists these key events.
Okay, let's walk through the big types of injury. You mentioned lack of oxygen.
Right. Ischemic and hypoxic injury. Hypoxia just means not enough oxygen. And the most common cause of hypoxia is ischemia-reduced blood flow.
So less blood flow means less oxygen delivery.
Precisely. And this kicks off a critical cascade. Imagine, like figure 4.8 in the text shows, no oxygen means the mitochondria can't make enough ATP. Without ATP, vital pumps like the sodium-potassium pump fail. Sodium rushes in, water follows.
And the cell swells up.
Exactly, cellular swelling. And other things start to fail too. Protein synthesis machinery detaches, for example. If oxygen isn't restored fast enough, this swelling progresses, the membrane gets leakier, and eventually, boom, irreversible injury. Think heart attack, stroke, classic examples of ischemic injury.
But you mentioned restoring blood flow can sometimes cause more injury.
Yeah, it's a paradox called ischemia-reperfusion injury. When blood flow and oxygen suddenly return to tissues that have been ischemic for a while, it can trigger a new wave of damage. Figure 4.9 outlines this.
How does that happen?
Several things contribute. A burst of reactive oxygen species oxidative stress. Calcium levels inside the cell can get overloaded. There's inflammation, immune cells rushing in. It's complex, but the restoration itself can be injurious.
Okay, you mentioned reactive oxygen species, ROS. This oxidative stress sounds important. Let's dive into that because it seems to pop up everywhere.
It really does. So a free radical is basically an unstable molecule with an unpaired electron. It's desperate to become stable, so it reacts aggressively with nearby molecules, lipids and membranes, proteins, even DNA stealing an electron and causing damage in the process.
Where do these come from?
Normal metabolism generates some, but also things like UV light, radiation, inflammation, or metabolizing certain chemicals or drugs. Table 4.3 lists some specific ones. The damage they cause, as shown in figure 4.11, includes breaking down cell membranes, altering proteins so they don't work right, causing DNA mutations. They can really mess things up, especially mitochondria.
But we have defenses, right? Antioxidants.
Absolutely. Our bodies make enzymes like superoxide dismutase, SOD, and catalase. We also get antioxidants from our diet. Vitamin C and E are classic examples. Table 4.4 lists quite a few. These guys neutralize the free radicals. But if the production of ROS overwhelms the antioxidant defenses...
Oxidative stress.
You got it. And that's linked to a whole host of problems. Atherosclerosis, Alzheimer's, cancer, diabetes, just aging in general. Box 4.2 highlights these connections.
Okay, moving on. What about direct chemical or toxic injury?
Right. Exposure to xenobiotics, foreign chemicals. This could be anything from industrial chemicals to pollutants, pesticides, even some plant or microbial toxins. Your liver is often on the front line here trying to detoxify these things.
The first pass effect.
Exactly. Figures 4.12 to 4.14 illustrate how the liver tries to transform these chemicals, but sometimes the process itself creates harmful byproducts or the liver just gets overwhelmed. Think about drugs too. Things like arsenic or cyanide are direct poisons. Even common drugs like acetaminophen can be toxic in overdoses. And sadly, the opioid crisis highlights the devastating impact of drug toxicity. Tables 4.5 and 4.6 show some grim U.S. stats.
Environmental toxins are a huge issue, too. Air pollution.
A massive one. The book calls it the world's largest single environmental health risk. Tiny particles like PM2.5 get deep into your lungs, causing inflammation, cardiovascular issues. Figure 4.15 and 4.16 show sources and effects. Millions of premature deaths worldwide.
And heavy metals, like lead.
Still a concern, especially lead in old paint. Kids are particularly vulnerable. They put things in their mouths, their brains are developing rapidly, their blood-brain barrier isn't fully formed. Lead is neurotoxic. Table 4.7 lists others like mercury, arsenic, cadmium.
Let's talk about ethanol alcohol.
Ah, yes. Alcohol metabolism produces acetaldehyde, which is toxic and a carcinogen. Chronic heavy drinking has major effects. Nutritionally, it can lead to deficiencies in things like magnesium, vitamin B6, thiamine, folic acid. That folic acid deficit is especially bad in pregnancy, contributing to fetal alcohol spectrum disorders. FASD figure 4.18 shows some characteristics.
And the organ damage.
Significant. CNS depression, obviously, but also alcoholic liver disease, starting with fatty liver, see figure 4.17, progressing potentially to inflammation, steatohepatitis, and finally irreversible scarring, cirrhosis. Also cardiomyopathy, pancreatitis, increased cancer risk. It's systemic.
But what about that J-curve thing where moderate drinking seems linked to lower mortality?
Yeah, the data suggests that, compared to non-drinkers, moderate alcohol consumption might correlate with slightly lower mortality from some causes, particularly cardiovascular. But it's a fine line, and heavy drinking is unequivocally harmful across the board. It's a complex picture.
Okay. Then there are unintentional and intentional injuries, physical trauma.
Right. The U.S. statistics mentioned are sobering: drug poisoning, car accidents, firearms are major causes of injury death. The book categorizes them. Blunt force injuries cause things like contusions, bruises. Remember you asked about the color change?
Yeah, red to blue to green to yellow.
That's hemoglobin breaking down. Figure 4.22 shows it. Red-blue is hemoglobin, green is biliverdin, yellow is bilirubin, brown is hemosiderin. It's visible pathophysiology. Blunt force also causes lacerations, tears, and fractures. Table 4.8 lists these.
And sharp force.
Incised wounds longer than deep, stab wounds deeper than long, puncture wounds, chopping wounds. Each leaves characteristic marks. Gunshot wounds too, entrance versus exit, range of fire, all detailed in Table 4.8.
What about asphyxial injuries? Lack of oxygen again, but different causes.
Exactly. It's about cells failing to receive or use oxygen. Suffocation could be lack of oxygen in the air, like in a confined space, or the airway being blocked. Strangulation involves compression of the neck structures. Interestingly, it takes less pressure to block the jugular veins than the airway itself.
And chemical asphyxiants.
We touched on carbon monoxide, CO, odorless, colorless, binds hemoglobin like crazy, causes that cherry red skin. Cyanide stops cells from using oxygen at the mitochondrial level, sometimes has a bitter almond smell, though not everyone can detect it. Hydrogen sulfide, rotten egg smell, also interferes with oxygen utilization. Drowning is another form, causing hypoxemia.
Wow, okay. Briefly, what else causes cell injury?
Well, infectious injury from microbes, immunologic and inflammatory injury where your own immune system causes damage, genetic factors like in sickle cell disease, nutritional imbalances, either deficiency or excess, and physical agents, temperature extremes, radiation, noise, mechanical stress. Table 4.9 lists a bunch of these miscellaneous factors.
So cells adapt, they get injured. How does this injury actually look or manifest itself, besides systemic things like fever?
A key way is through intracellular accumulations or infiltrations. Basically, cells start storing stuff they shouldn't, or too much of something normal.
Like hoarding.
Kind of. Figure 4.19 shows 4 main ways this happens. Maybe the cell can't remove a normal substance fast enough, like fat in the liver, or there's a genetic defect causing an abnormal substance to build up, or the cell lacks an enzyme needed to break something down. Or, finally, some harmful external substance gets stuck inside, like carbon particles.
What are the common things that accumulate?
Water is probably the most common cellular swelling or hydropic degeneration. We saw that with hypoxic injury due to Na plus K plus pump failure. Figure 4.20 shows these swollen kale cells. It's often reversible early on.
What about fats?
Lipids and carbohydrates can accumulate in various storage diseases, but the big one is steatosis, or fatty liver. Figure 4.21 shows liver cells just packed with fat droplets, super common with alcohol abuse, diabetes, obesity. Cholesterol accumulation is key in atherosclerosis, those fatty plaques in arteries.
Proteins.
Pigments. Proteins can build up, sometimes abnormally folded proteins, which are implicated in diseases like Alzheimer's, neurofibrillary tangles, or alpha-1 antitrypsin deficiency leading to emphysema. Pigments can be endogenous, made by the body, like melanin, skin color, or hemosiderin, iron storage seen in bruises. Bilirubin accumulation causes jaundice, or exogenous pigments from outside, like carbon in miners' lungs or tattoo ink. Figure 4.22, the bruise, shows hemoprotein pigment changes.
And calcium.
Calcium accumulation is interesting. Dystrophic calcification happens in already damaged or dying tissues. Think gritty deposits in old scar tissue, damaged heart valves, figure 4.24 shows this, or atherosclerotic plaques. It's a sign of previous injury. Metastatic calcification, on the other hand, happens in normal undamaged tissues, but it's caused by high levels of calcium in the blood, hypercalcemia. This might be due to things like overactive parathyroid glands or certain cancers. Figure 4.23 relates this to disturbances in calcium balance.
One more accumulation, urate.
Right, urate or uric acid. Too much leads to crystals forming, typically in joints, causing the intense pain of gout.
And you mentioned systemic signs earlier, fever, faster heart rate.
Yes, table 4.1 lists these. Fever, increased heart rate, leukocytosis, high white blood cell count, pain, and importantly, the release of intracellular enzymes into the blood when cells are damaged. Measuring enzymes like LDH, CK, AST, ALT tells doctors about potential organ damage, heart, liver, muscle, etc.
Okay, this is where it gets really fundamental, right?
Yeah.
Irreversible injury leads to cellular death.
The final stage for an injured cell. Traditionally, we talked about necrosis and apoptosis. Our understanding is evolving, recognizing that even necrosis can sometimes be programmed, a process called necroptosis. But the core distinction shown in figure 4.25 and table 4.11 is helpful.
Let's start with necrosis. That's the messy one.
Generally, yes. Necrosis involves the sum of changes after local cell death, including self-digestion, autolysis. The key is loss of membrane integrity. The cell basically bursts open, spilling its contents and triggering significant inflammation. The nucleus undergoes characteristic changes: shrinking, pinosis, fragmenting, karyarexis, and finally fading away, karyolosis.
And there are different types of necrosis. Figure 4.26 shows a few.
Right. Coagulative necrosis is common in solid organs like the heart or kidneys after ischemia. Proteins denature and become solid, opaque, think cooked egg white. The tissue structure might be preserved initially.
Sure.
Liquefactive necrosis happens typically in the brain after ischemic injury because brain cells are rich in digestive enzymes. It also happens in bacterial infections. The tissue literally liquefies.
Yeah.
Caseous necrosis is characteristic of tuberculosis. It's a combination, partly coagulative, partly liquefactive. Dead cells disintegrate but aren't fully digested, leaving a crumbly, cheesy material within a granuloma.
And fatty necrosis.
This involves enzymes called lipases breaking down fat, usually in fatty tissues like the breast or around the pancreas. This releases fatty acids that combine with calcium to form soap-like deposits, saponification. The tissue looks chalky white.
What about gangrene? Is that a type of necrosis?
Not exactly a specific type of cell death, but rather necrosis affecting a large area of tissue. Dry gangrene is usually due to coagulative necrosis from severe hypoxia, often in a limb. The tissue becomes dry, shriveled, dark brown, or black.
And wet gangrene.
That's much worse. It involves liquefactive necrosis, usually complicated by bacterial infection. The tissue's cold, swollen, black, and has a foul odor due to bacterial action. It can spread rapidly and be lethal. Gas gangrene is a specific type of wet gangrene caused by Clostridium bacteria, which produce toxins and gas bubbles in the tissue. Extremely dangerous.
So necrosis is messy, inflammatory death. What's the alternative?
Apoptosis. Yes, apoptosis is programmed cell death. It's an active, highly regulated process where the cell essentially triggers its own destruction in a neat, contained way. Think of it as cellular suicide, but well organized.
Is it always bad?
Not at all. It's crucial for normal development and tissue turnover. Your body eliminates billions of cells every day via apoptosis to maintain balance. But it can be triggered pathologically, too.
Like when?
If a cell is severely injured beyond repair, or if misfolded proteins accumulate in the cell, this happens in neurodegenerative diseases like Alzheimer's, as shown in figure 4.27. Viral infections can trigger it as part of the immune response. Dysregulated apoptosis is also a problem. Too little can contribute to cancer or autoimmune diseases, while too much can cause tissue damage in conditions like stroke or neurodegeneration.
How does it work? Is it different from necrosis?
Very different. It involves specific enzymes called caspases that activate a cascade, dismantling the cell from within. There are two main pathways shown in Figure 4.28, one triggered internally, mitochondrial pathway, and one by external signals, death receptor pathway. Crucially, the cell shrinks, breaks into small membrane-bound fragments called apoptotic bodies, which are then tidally engulfed by phagocytic cells. Minimal leakage, minimal inflammation. Very clean compared to necrosis.
There's one more process mentioned, autophagy. What's that?
Autophagy means eating of self. It's like the cell's internal recycling program, described in box 4.3. The cell basically wraps up portions of its own cytoplasm, including damaged organelles or misfolded proteins, in a membrane vesicle, and delivers it to lysosomes for breakdown and recycling. Figure 4.29 shows this process.
Why would it do that?
It's a survival mechanism during stress, especially nutrient deprivation. The cell can cannibalize itself to generate energy and building blocks. It's also critical for removing damaged components, like worn-out mitochondria, keeping the cell healthy.
So it's important.
Hugely important. Problems with autophagy are implicated in cancer, heart disease, neurodegeneration, and it seems to become less efficient as we age, which might contribute to age-related cell damage because the cleanup crew isn't working as well.
This naturally leads us to the bigger picture, aging and somatic death, the end of the line for the whole organism.
Right. Aging itself is seen as a normal, inevitable process involving a gradual decline in our ability to maintain that crucial homeostasis. It's a complex genetics, inflammation, oxidative stress, metabolic changes, all play roles.
And there's a difference between how long we can live and how long we do live, right?
Exactly. Lifespan, the maximum potential, maybe 80, 100 years, hasn't changed much.
Yeah.
But life expectancy, the average number of years remaining, has increased dramatically due to public health, better nutrition, medicine. Box 4.4 discusses this. The real goal now, many argue, is extending health span, the years lived in good health. Table 4.12 details typical degenerative changes with age.
And sometimes older adults experience frailty.
Yes, frailty is a specific clinical syndrome. It's more than just aging. It involves vulnerability, low energy, weakness, slow walking speed, unintentional weight loss, increased risk of falls, disability, death. Figure 4.30 shows it's multifactorial, involving declines across multiple physiological systems. Things like muscle loss, sarcopenia, bone thinning, osteopenia, anemia are often part of it.
Finally, the death of the entire organism, somatic death.
When the body as a whole ceases to function, the postmortem changes that follow are predictable and, importantly, don't involve inflammation because the body's systems have shut down.
What are those changes?
There's a sequence. First, pallor mortis, the skin becomes pale. Then, algor mortis, the body cools down gradually. Rigor mortis sets in after about 6 hours as muscles stiffen due to lack of ATP, peaking around 12 hours, then resolving. Livor mortis is the pooling of blood due to gravity, causing bluish-purple discoloration visible after a couple of hours, helpful for forensics. Then putrefaction, bacterial breakdown causing bloating, discoloration, odor, starting 24-48 hours after death. This leads to decomposition, breaking down organic matter, and eventually skeletonization.
Wow. Quite the journey from a single cell adapting to the fate of the entire organism.
So reflecting on all this, what's the big takeaway?
I think it's the incredible complexity and, honestly, elegance of these processes. How cells strive to maintain balance, how they respond to injury in predictable ways, and how even death at the cellular level is often a highly regulated, programmed event.
So considering that precision, the adaptation, the injury response, programmed death, makes you wonder, doesn't it? It really does. How could a deeper grasp of these fundamental mechanisms help us not just treat diseases after they happen, but maybe proactively enhance our health span? Prevent some of this injury cascade before it starts? What does understanding this intricate cellular dance make you think about regarding your own health and longevity?
Definitely food for thought. A fascinating look at the foundations of pathophysiology. Thank you for joining us on this deep dive into altered cellular and tissue biology. Keep exploring, stay curious, and we hope you'll join us again next time.